Hello everyone, welcome to Bioworks. So our topic for today is development of embryo in plants. So let's begin. So let us understand development of dicot embryo. So I have shown a diagram here which shows various stages that are involved in development of an embryo, and here I am specifically talking about a dicotyledonous embryo. As we all know, in a dicot embryo there are two cotyledons present. So we are going to see dicot embryo first, and then I will move to the monocot embryo. So starting with the zygote first, as we all know, in the process of double fertilization a zygote is formed, and this zygote then starts dividing, and here you can see I have shown two cells that are formed after the division. Now this stage is called as a proembryo. An embryo with two cells is called as a proembryo, and the cell that is towards the micropylar end is larger and is called as a basal cell, also called as a suspensor initial cell, and the smaller cell towards the chalaza is called as a terminal cell or embryonal initial cell. Alright, now for convenience, everywhere I have mentioned M and C, which indicates micropylar end and the chalazal end. Okay, coming to the next step now, if you can see, the basal cell or the suspensor cell starts to divide, and simultaneously the embryonal cell also starts to divide. I will right now focus only on the suspensor cell. So you can see the basal cell or the suspensor initial cell undergoes division. Here you can see two cells followed by formation of a row of 6 to 8 cells, right? Now the cell towards the micropylar end, the cell of suspensor that is towards the micropylar end, gets swollen and functions as a hostorium. I have shown that over here, right? Yes, it functions as a hostorium. And the cell that is towards the lower side, this cell, the last cell of suspensor, is called as hypophysis. Now, what is the role of suspensor here? This entire suspensor anchors the embryo to the endosperm. Right? This is the embryo. This is the part that is going to give rise to the embryo. So what is the role of suspensor shown in red here? It anchors the embryo to the endosperm, also kind of pushes the embryo towards the endosperm so that the embryo can absorb nutrients from it. Right. Now that was about suspensor. Let us now look into the terminal cell I was talking about. Now the terminal cell or the embryonal initial cell undergoes division. Right, you can see the two cells that are formed here, and then further they will undergo second division to form a quadrant. Right, you can see the 4 cells formed here, which we call it as a quadrant, and quadrant stage is followed by an octant stage because there are total 8 cells that are formed. An important thing to note here is the cell division that occurs in the embryonal cell. All the cell divisions are right angle to each other up to the formation of an octant. Now after the formation of octant, you will see the first stage of the embryo form, which is called as a globular embryo, because here a globular shape is achieved. Right, you can see the shape of the cells of the embryo proper has taken up a globular shape, right? And this is the suspensor towards the micropylar end. I have labeled suspensor over here, right? Now hypophysis, which I mentioned earlier, hypophysis further divides to give rise to radicle and the root cap. I have shown radicle at the final stage here, but the cells of the radicle start to form as early in the globular stage itself. Now, followed by the globular stage is the heart-shaped embryo. So how is the heart-shaped embryo formed? What happens is the cells of the embryo in the globular stage of embryo, they start giving rise to hypocotyle and cotyledons. So because of initiative growth of the cotyledons, this heart-shaped is achieved. You can see the embryo looks heart-shaped here, right? And then followed by the heart-shaped, when the cotyledons start to grow further, they will achieve the torpedo-shaped embryo. Here you can see how the embryo looks like a torpedo shape, or you can also call it as a horseshoe shape. Right, and here in the torpedo shape, that is the mature embryo, I have labeled radicle, which is formed from the hypophysis. There is plumule here, and these are the two cotyledons. Now since we are talking about a dicot embryo, here I have shown two cotyledons. Right, so that was all about development of a dicot embryo. Let us now understand development of a monocot embryo. Now, we have already studied development of a dicot embryo. So, the stages that are involved in development of a monocot embryo are quite similar. If you see the first stage over here where the zygote divides into two cells called as a proembryo, and the proembryo has a suspensor initial cell and an embryonal initial cell. These cells further give rise to a suspensor. You can see a suspensor is formed which pushes the embryo towards the endosperm for nutrition, and this is the embryo proper, right? So right from the proembryo stage up to the octant stage, the steps of development of monocot embryo remain similar to that of a dicot embryo. Now after that, two important things to note over here is you can see the cotyledon that is formed, it is single and it occupies a terminal position. That is one important thing to note down. And second important thing is the plumule is lateral. You can see the plumule is towards the lateral side. Alright. Now cotyledon later modifies to something called as scutellum. Now what you can see in this figure, this is an LS of an embryo of a grass. Right. So scutellum over here is nothing but a cotyledon. And you can see all these labels. I'm going to explain them. Over here, shoot apex gives rise to a plumule. Plumule, as we all know, plumule forms the shoot of the plant, right? And the protective covering outside the plumule is called as coleoptile. Then similarly, you have a radicle over here, which gives rise to the root later on. And then you also have a root cap. And then the protective sheath covering present around the radicle is called as coleorhiza. So remember these two terms, coleoptile and coleorhiza. And apart from these, there is also something called as epiblast, which is like a flap-like structure over here. It is said to be like a remains of cotyledon in monocots. Alright, that was all about development of monocot embryo.
So hope the video was useful to you all. See you in the next session. Thank you for watching.